Hello everybody. Caffeine is a common chemical that we interact on a daily basis, and we can have access of it in many forms. Today we are going to be looking at the determination of concentration of caffeine of a specific black tea brand.
The details on the packaging indicates that it should contain 11 mg of caffeine per 100 g of dry tea leaves. Is it the particular detail accurately presented? Is it true?
So for this analysis, we will be using HPLC, also known as high performance or pressure liquid chromatography. And that is what we are looking into today.
HPLC was first developed during the 1960s by Professor Chaba Harvard as a direct offshoot by the classic column liquid chromatography. Through the improvement in the technologies of the columns and instrumental components such as pump, injection valve, and detector.
HPLC is used for identifying, quantifying, and purifying the individual components of the mixture. Reverse phase HPLC can be used to determine the amount of caffeine in several beverages. In this experiment, we will be determining the amount of caffeine in black tea. In reverse phase HPLC, the stationary phase is non-polar adsorbent, whereas mobile phase will consist of a polar solvent. In this analysis, C18 column will be the stationary phase, while water mixed with methanol will be used as the mobile phase.
The illusion in this HPLC is isocratic since the mobile phase is constant throughout the whole analysis. There are few components in the HPLC which are important. Firstly, the HPLC pump functions to deliver the mobile phase through the system in a controlled, accurate, and precise manner. Then, we have the column which is usually constructed of stainless steel tubing. It separates the sample components of interest using various physical and chemical parameters.
Other than that, we also have the detector. The detector translates the sample concentration changes in the HPLC column effluent into electrical signals. The choice of the detector used depends on the solute type and concentration, detector sensitivity, linear range, and compatibility with the solvent and elution mode to be used, as well as cost.
The effluent flows through a flow cell and is analyzed by the detector system. These flow cells are delicate and easily polluted or damaged. Hence, it's very important to filter the mobile phase before the analysis. In this experiment, the photodiode array detector is used as it is able to read multiple wavelengths simultaneously by a microprocessor to provide a spectrum of 200 nanometer to 700 nanometer every 0.1 second.
So, how to perform HPLC? A blend consisting of only the mobile phase, 3 serial dilution as 3 ppm, 6 ppm, and 9 ppm for caffeine standard solution were prepared and placed into each volumetric flask. For the sample preparations, 2 sets of samples of 0.5 ml of black tea was pipetted and made it to the volumes of volumetric flask by adding mobile phase. Trace filter was used while transferring the sample into the vials. This practical uses reverse phase whereby the stationary phase is non-polar C18 columns, while the HPLC is polar, which is a mixture of methanol and water. Phosphodiode array detector was used as a detector. The HPLC setup was done by the lab officer prior to this experiment. The stock solution and sample were injected accordingly on the tray. Since the injection is automated, the solution on the tray were injected into HPLC system automatically. Repeat step 5 for the 5 standard dilution and sample dilution. The retention time and peak areas of the standard dilution were recorded and a standard curve was plotted. Retention time and peak area of sample were recorded and the concentration of the sample was calculated by extrapolating the curve. So here is a summary of our procedure.
These are the results that we obtained from the HPLC analysis. The concentration of the sample is obtained by extrapolating the graph. By using the equation that is given, the concentration of the sample is 6.892 ppm. After the calculation, the amount of caffeine in 100 gram of tea sample is 34.46 milligram.
Reverse phase chromatography is when the stationary phase used is a non-polar adsorbent and the mobile phase consists of a polar solvent. In this chromatography, solutes are retained due to the hydrophobic interactions with the non-polar stationary phase and are eluted out in order of increasing hydrophobicity or decreasing polarity. 2 samples of black tea were used to get an average value for a more accurate value of peak area. The reason why we use the blank is because without the blank, the y-intercept obtained will be inaccurate and this will affect the final value of caffeine that we will obtain. However, the blank can be neglected in this case as it does not give any peak area or retention time.
The retention time obtained for all the standards were in the range of 2.120 to 2.126 minutes. The average retention time obtained was 2.125 minutes. Hence, it is clear that the retention times of the black tea samples are very similar to the retention time of the standard stocks used. This proves that the sample matches with the standard solutions used. The peak area obtained here indicates the amount of caffeine present in the black tea. Based on table 1.0, the lowest peak area was obtained by the caffeine standard concentration at 3 ppm, whereby the peak area was 34871 AU, followed by 6 ppm and 9 ppm. According to Peirce's law, the peak area and concentration are directly proportional, says Torsi et al. 1989. As the concentration increases, the peak area increases as well. As we can see, based on the standard curve, that is what we have obtained.
To compare with the label on the pack saying there is 11 mg of caffeine per 100 gram of dried tea leaves, the amount of caffeine that we obtained is higher and the label is not accurately presented. According to Goodwin 2019, brewing methods and styles can have a large impact on a tea's caffeine level. By using a higher water temperature, longer brewing time, or a higher ratio of tea leaves to water, these factors will affect the caffeine level by increasing the level of the brew. Besides, by using tea bags, it also can influence the tea's caffeine level.
The advantages of using HPLC is, it is a quick, automated, and highly accurate method to identify certain chemical components in a sample. The entire process can be completed within 30 minutes. Instead of using gravity, a pump is used to force liquid solvent to flow through a solid absorbent materials. Different materials will be separated according to their own speed. As HPLC is usually automated, hence it can be run with minimal trainings.
Besides, HPLC is versatile and extremely precise when it identifies and quantifies chemical components. As the processes are normally automated, many steps have been reduced and hence the precision and reproducibility of analysis will increase.
However, co-elution will happen when analyzed the sample by HPLC. Compounds that have the same polarities and structure will be eluted out almost at the same time. This causes the peaks formed will be nearly the same. Hence, it is difficult to determine the exact components eluted from the sample mixture when co-elution happens.
Other than that, the elution of analytes will be affected when the sample mixture contains a high binding strength of analytes. Different analytes from the mixture will bind to the column in different strengths. Some of the compounds will bind to the column with very high strength. They will not be released from the beads in the column and will be eluted and detected throughout the analysis.
Thank you. Bye.